Welcome to this poster on the strengths and limitations of causal inference for gene regulatory networks in yeast. I'm Adriaan Ludl of the Computational Biology Unit at the University of Bergen in Norway. This poster is based on work recently published in Molecular Omics. You can follow the analysis pipeline in the GitHub repository Finder Causal Network Inference on yeast. Our work used data from two papers. One is an eLife paper published in 2018 entitled Genetics of Transregulatory Variation in Gene Expression, and the other is the ground truth data from the yeast-tract database. We have used the causal inference tests implemented in Finder, which is also an open source tool available on GitHub. And it implements a correlation-based test and 5 causal tests, for which the causal models are shown on the table on the right. On the left-hand side, you can see the matrix of interactions in the yeast genome predicted by the correlation test, and this can be compared with the matrices for the two causal tests. Here I show the instrumental variable test P2 and the mediation test P2, P3 as examples. And we see that in both P2 and P2, P3, there are vertical bands appearing, which are not visible in P0, the correlation-based test, which is essentially a symmetric matrix. But in P2 and P2, P3, we see that there are, at certain positions along the genome, regulating genes that have targets that range across the whole yeast genome. We have validated our predictions using the yeast-tract ground truth interactions. Here I show the precision and recall curve for the DNA binding and expression ground truth from yeast-tract, and we see that several methods perform well to varying degrees. And in the inset, we see the fold change in the area under the precision recall curve for 5 tests: P2, P3, P2, P2, P5, P, and P0. P0 is the correlation test and performs worst. 3 instrumental variable methods perform best, and the P2, P3 method performs also quite well in this general metric. Now, we wanted to discern the effect of sample size on the predictions. So we've done subsampling on our data, and we see that at low sample sizes, the methods do not perform very well, but at larger sample sizes, the causal methods perform significantly better than correlation-based ones, which stagnate at a low AUPR fold change. And also we see that the mediation-based method has a stagnation in its performance between 400 and 1000 samples, whereas the trend towards improved predictions seems consistent for the instrumental variable methods. In this figure, I show the target counts for the regulating genes. So regulating genes are shown according to their ordering in the yeast genome and the chromosomes along the x-axis, and the target counts are the vertical height of the bars. Panel A shows target counts for P2, P3, a mediation-based method, and in our paper in Molecular Omics, we discuss several genes for which we have very high target counts. In panel B, we show the predictions for the instrumental variable method P2, as well as the hotspots from the Albert et al. Paper. And for certain of these hotspots, we have also predictions shown in red, which we can compare. In particular, our paper discusses how P2, P3 performs well for DNM1, but fails for STB5, and I invite you to look into the paper or to come to the discussion in the virtual poster session. Finally, panel C shows the genotype covariance matrix, and this is shows a certain reflection of the bands seen in P2, which are the gray peaks in panel B. With this, I would like to conclude. Finder allows us to efficiently compute and test causal models to obtain context-specific causal gene regulatory networks. And the mediation and instrumental variable methods perform better than correlation. More specifically, instrumental variable methods display a higher sensitivity and mediation methods show a higher specificity, but the performance of mediation worsens with increasing sample size. We are working to address the following limitations. The first one is related to the data where shared EQTLs or genetic variants occur. There were 337 EQTLs that are strongest for 2 genes and a handful that were stronger for even a larger number of genes. In addition, the instrumental variable methods are susceptible to false positive predictions due to genomic linkage and pleiotropy, and we are working on improving our methods to avoid this issue. Thank you for listening, and I hope to see you in the virtual poster room, which you can find under the tab Poster Room on the conference webpage.